Okay. Bismillahirrahmanirrahim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Allahumma salli ala Sayyidina Muhammad wa ala alihi wa sahbihi ajma'in. Respected Madam Yusrina Abu Bakar, covering Chief Librarian Dar Al-Hikmah Library IIUM, our respected panelists for today, Professor Dr. Yumi Zuhanis, Hashim from International Institute for Halal Research and Training INHART, and Associate Professor Dr. Muhammad Ibrahim from Kuliah of Allied Health Sciences, academics and researchers, library top management, senior management, head of units, and colleague from Dar Al-Hikmah Library IIUM, officer within IIUM and without. Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh and very good morning. My name is Nurul Zahra binti Johari and I'm your moderator for today. To gain barakah of our event, let us start with lafaz Bismillahirrahmanirrahim and let's make dua to Allah to bless our event and hope our session today will be beneficial to all. On behalf of Dar Al-Hikmah Library, we wish to extend our warm welcome and thank you to our special guests and all participants to this session on Research Fast Talk, Expert in Action, Series 1 for year 2022. Okay, ladies and gentlemen, as IIUM is heading towards responsible research and innovation and humanizing education, researchers started to perform more impactful researches which are beyond academia. Thus, the main objective of this session is to provide a platform for the researchers to share their research to the public. Hopefully, all of us will be able to reap as many fruitful inputs and tips from this session, inshallah. Ladies and gentlemen, we are very honored today to have our invited panelists to share their research experience with us within 40 minutes each. And while listening to them, you are free to post the question in the chat box, and inshallah, we will respond later during the Q&A session. Okay, now it is my pleasure to introduce our first panelist for today, Professor Dr. Yumi Zuhanis Hashim. Assalamualaikum, Prof.
Waalaikumsalam warahmatullahi wabarakatuh.
How are you, Prof?
Alhamdulillah, good today.
Okay, Alhamdulillah. I think all participants are very excited about today's topic. Same as me because Gaharu, we normally know Gaharu, but we don't know the in-depth about Gaharu. So inshallah, we are looking forward for today's topic. Okay, so let me read briefly the biodata of Prof. Yumi. Okay, Prof. Dr. Yumi Zuhanis Hashim is currently the Deputy Dean Academic and Internationalization and Professor at the International Institute for Halal Research and Training INHART. She had previously served the Department of Biotechnology Engineering, Kuliah of Engineering IIUM since 2001. She held several administrative posts at Halal Industry Research Center and contributed to its development and then the establishment of INHART. In 2018, she fully transferred to INHART. She has more than 20 years of teaching experience in the area of natural product, phytochemical processing, mammalian cell culture technology, halal science, and halal studies. Dr. Yumi graduated with Biomedical Science from University Kebangsaan Malaysia in 1999 and Master in Bioprocess from University Technology Malaysia in 2002. She worked as a research officer at the Chemical Engineering Pilot Plant, now known as Institute of Bioproduct Development, University Technology Malaysia, before joining the academia. She then obtained her PhD from the Faculty of Life and Health Sciences, University of Ulster, UK, and served as a postdoctoral research fellow at the Institute of Food and Health, University College Dublin, between February 2007 until December 2008. Dr. Yumi also is an active researcher in natural products and is interfaced with health benefits and halal living, with special interest in Agarwood, halal science, and halal studies. She has received grants from government agencies as well as industries. She has published more than 40 journals articles, several book chapters, and presented her research output in various local and international conferences. She is actively involved as editorial member and serves as reviewer in various peer-reviewed journals. She also sits as expert panel member in several national level committees, including Academic Program Standard and National Fund Review Committee. Dr. Yumi also is a fellow of Institute of Biomedical Science UK and an alumni of Young Scientist Network Academy Science Malaysia. Okay, and now presenting to you, ladies and gentlemen, Professor Dr. Yumi Zuhanis Hashim to share her research project with us entitled 10-year Agarwood Gaharu research journey from Maqasid-based bioprospecting to development of Halalan Toyyiban product. Please welcome, Prof.
Thank you, Madam Moderator, Sister Zara, for the kind introduction. Alhamdulillah Rabbil Alameen, wassalatu wassalamu ala ashrafil anbiya wal mursalin, wa ala alihi wa sahbihi ajma'in. Assalamualaikum warahmatullahi wabarakatuh to everyone today. First of all, let us thank Allah for His blessing that we can be here today in this sharing session. And I would also like to thank the IM Dar Al-Hikmah Library for inviting and giving me the opportunity to share some of our research journey, some stories from our research journey. So the title of today's sharing would be 10-year Agarwood or Gaharu research journey from Maqasid-based bioprospecting to development of Halalan Tayban products. So the sharing session today will be outlined as such. First, it will be into the introduction of Agarwood, and then a bit on our work on Agarwood as source of health beneficial compounds. And then we will try to share some of the way forward of Agarwood research as well as for general natural products. So the first one is introduction to Agarwood. I would like to bring your attention to the photo of the Agarwood tree. So if you have not seen the Agarwood tree before, the photo shown to you just now is actually in our IIM compound. So if you would like to see that real Gaharu tree in our IIM compound, you can go to our Kuliah of Engineering E5 in the courtyard. So in the beginning, we had planted 4 Gaharu plants there, and then another colleague of us planted another 4 from a different species. So maybe it's still there because of pandemic. I have not been there as well, but I hope it's still thriving there. So everyone who is interested to see real life Gaharu tree, you can always go there. Okay, now let's move on to introduce to you the Agarwood from various languages. Of course, Agarwood is most famous for its aromatic resin. I think many of us know about that. The aromatic scent of its wood-infiltrated resin and oil. So we call it kayu gaharu in our native language, Malay. But of course, it's also well known as oud in Arabic, chenxiang in Chinese, ginkgo in Japanese, agar in Hindi, and even in Malay, some of us also know it as karas, pokok karas. From scientific point of view, Agarwood belongs to the Thymelaceae family. This is a family, and the aromatic resin infiltrated Agarwood has been found in several genera of this family, namely Aquilaria, Gonistylus, and Girinops. And our team works on the Aquilaria species, particularly on the Aquilaria malacensis, which is endemic in Malaysia. So malacensis, if you can connect it, it sounds like Malacca. So that is from our endemic species. And also we work on Aquilaria subintegra, which is believed to be originated from Thailand. However, other popular species under investigation by Agarwood research community includes A. sinensis, which is very much related to China, as well as A. krasna. For researchers in Thailand, they usually use this species. And another species is Agalocha, which is normally being researched by our researchers in India. This slide shows you the overall bioprospecting aspect of Agarwood. So we know that it is very popular for its use in perfumery industry. But if you can see from in the upper left-hand side of the slide, there are also not so much popular purposes of Agarwood. So it's used as sculpture as well in various cultures. Some of it used as rope. On the lower side, on the left-hand side, the lower side is where we put in the traditional medicinal practice of people, especially in the Asian region, that we have been using this agarwood as part of the traditional medicinal practice. So interestingly, the unique profile of the compounds also has been the underlying factors where this unique agarwood compound has been used in this traditional medicinal practice of various researchers. But in the modern world, we are actually using this bioprospecting approach that we try to get the best benefit of the agarwood tree and produce or develop into different kind of products, which is, you can see here, like nutraceuticals, pharmaceuticals, cosmetic products, complementary alternative medicine, and all of those products are actually, we want to make them into Halalan Toyyiban products. As per slide before, here I'm trying to show you where agarwood is in our traditional medicines. It is not just in the well-established, well-known Chinese traditional medicine which is being practiced. It is also there in Ayurveda, it is also there in the Japanese traditional medicine, also there in the Korean, also there in the Unani, and of course also there in our Malay traditional medicine. So that is just some of the say recipe or formulation which we can see from the kitab tip. Okay, this slide is a bit busy. So what I'm trying to tell here or share with everyone is that when we say Gaharu or agarwood, normally we would refer to the resin, to the wood, which is the aromatic wood. However, Gaharu also is referred to the plant itself, as you can see from this slide here, the same plant that I've shown you in the earlier slide. So from this plant, you can see there are many raw materials that can be a starting material towards the bioprospecting journey. So on the left-hand side is the uninfected one, and on the right-hand side is the infected one. How is this so? Because Gaharu or agarwood is very unique. So I think many of us will also know that the Gaharu, the resin itself, the aromatic wood itself actually only being developed in trees which are so-called disturbed. So what we mean by disturbed is, in the natural setting, disturbed perhaps could be the effect of lightning or the effect of the people living in the forest, the orang asli, who went to axe them to create some disturbance to the tree. While in the plantation, what we do normally is we inject them or we inoculate them in a systematic manner. But the objective of doing that is actually to disturb the immune system of the tree, thus the tree would produce the resin as part of its immune protection. So that product of immune system is actually the one that we are looking for normally in the research in the agarwood community. So if you can see on the left-hand side, there are many different types of raw materials that we can begin with. The flower, the plant, the fruits, the leaf, the white wood. The white wood we normally refer to the wood which is not being infected. And on the right-hand side, of course, after being infected, there are additional raw material that we sought for. Normally, we sought for the resin, as I was saying just now. But along that, we also have the byproducts. When we distillate the resin or the agarwood or the aromatic infiltrated wood, we would get the oil that we are looking for, as well as the byproduct, the so-called distillate, or some would also call it hydrosol. So all of these are actually potential raw materials for us to develop many different products as shown in the earlier slide. Okay, now let's move on to the second part of the sharing session. This is where I would like to share our team's work, the 10-year journey in Agarwood research. So our work on Agarwood actually focus on the health beneficial compounds. So in Agarwood research community, there are many scopes. It's a very big research community for Agarwood. There are some people working on agroforestry. There are some people working on agriculture scope of it. Some are focusing only on perfume and the compounds. Some are focusing on the inoculation and injection. But our team at IUM is actually focusing on the health beneficial compounds of many different types of raw materials as shown before. So this is some of the publications that we have contributed along the way, the 10-year journey. So I would like to spend a little bit time on this slide. So early on in 2010, 2011, we started with a focus on anti-cancer, looking at potential anti-cancer effects from the agarwood oil itself. However, because as many would know, the oil is very expensive. So we were challenged with the high cost as well as the inconsistent batch of materials coming from the industry. So to do a good research, we need to have the consistent batch. But that was the challenge back then, as well as the high price. Then we changed our strategy into using the so-called waste of the plantation or the byproducts of the agave plantation, starting with the white wood of the uninfected trees and then the leaf. So what happens in the plantation is usually they have systematic pruning. They cut off the branches, the small branches and the leaf in a systematic manner. So what we do was, we tried to work with industries, with the farmers, and we collect all this waste from the plantation and use it as our raw materials in our research. So as it takes several years for the agarwood to be mature, normally between 5 to 7 years, to be inoculated and then only produce the agarwood that we were looking for. So we had abundant supply of this raw material throughout the years. And during the early years as well, our work included a lot of extraction and optimization work and its relationship not only with the yield of extract, but more importantly, to see and maintain the desired biological effects. So this next slide are the more recent work done by our team, where we look into the anti-inflammatory effects on top of the anti-cancer effects, because in the cancer pathophysiology, actually anti-inflammatory is also one of the upstream activities. And recently as well, one of our team members, Dr. Hafiz, looked into the antibacterial effects of agarwood on the multi-drug resistant gram-negative pathogens. So we are also looking into this, and there are very promising results. So while doing this bioprospecting work earlier on, we adhere to the concept and principles of zero waste, waste to health, and of course the big umbrella of bioprospecting. And a bit later on, we embrace on the circular economy concept and the SDGs, particularly the SDG number 3 and SDG number 12. SDG number 3 is good health and well-being, and SDG number 12, responsible consumption and production. Although not much emphasis on that until very recently, where we are also embracing the university's advocation on these SDGs. But at this moment, 10 years ago, 5 years ago, there was no Maqasid-based yet, but inshallah I will inform you a bit later in the slides. One of the key highlights of our work is this publication in the Journal of Ethnopharmacology, published in 2016. So this was actually an output of my sabbatical leave. So after the sabbatical leave, alhamdulillah, we managed to publish this. And in this review, we looked at different materials used in agarwood research, particularly related to the biological effects. So if you can see on the lower right-hand side there, you can see that agarwood actually have many different, a myriad of biological effects, including anti-cancer, antioxidant, antimicrobial, and many others. More recently, we move into a more applied type of work. For example, here we successfully developed Agarwood leaf incorporated nanofiber patch. And this invention has won the Silver Award at the Malaysia Technology Expo, MTE 2020, and also has been patented. And another recent work from our PhD student who has also graduated recently looks into the development of agarwood leaf extract loaded nanocapsules, and these are being tested in zebrafish embryos towards use as anti-inflammatory therapeutics. So this one, we also work with CREAM, IIUM, on the zebrafish embryo work. And alhamdulillah, this has also been patented recently. Now, let's move on to the last part of this sharing session, which is the way forward for agarwood research and natural products. A quick check on Scopus has shown that research in agarwood started as early as 1959, and exponentially from the year 2007, we can see a growth there. In the early years, 1959, 1960s, the researchers were very interested into the compounds, the unique compound, the cis-terpenes in the agarwood resin itself. But along the way, you can see that the growth is actually comes in various scopes, as I mentioned before, either forestry, agriculture, perfumery, injection, inoculation, as you can see in the pie chart there. So in line with the growth of agarwood research, it is very important to put in deliberate efforts to uphold the sustainable principles of bioprospecting, as discussed previously, for a sustainable future of agarwood research. As I mentioned before, previously we got the materials from the forest itself. It was very unsustainable, and at one point the agarwood actually goes into a risk of extinction. So after that, the research or the agarwood community come together and put them together into this new way of doing things, which is the plantation. So that is why now we have a lot more abundant supply of raw materials for agarwood. So when we were working on the review paper in 2016 and until now, actually, we've seen some inconsistencies in use of terms in reporting agarwood-related work. So somehow it makes the agarwood research vicinity, sometimes they need to replicate unnecessary things. And so that is one of the challenges for researchers to actually adhere to responsible and ethical conducts, because when we need to repeat things that we should not be repeating, it means that we are using more resources. It also means more cost. So some of the things that we see and we would like to recommend for not only actually agarwood research community, but more generally to natural products research community, is the standardization of terms used in the research. Or specifically, for example, in agarwood research, because it's a complex development of the agarwood itself in the tree, some people will use different terms. For example, some people use infected versus inoculated versus artificial. Some would use uninfected versus inoculated versus healthy wood. So sometimes it's quite hard when you read a scientific paper to identify which part of the material, which material is actually the researcher is using. And even the Gaharu or the wood itself, the infiltrated wood itself, different researchers report it using different terms. Some would just use agarwood, as we are sharing it now. Agarwood mainly refers to the aromatic resin, but some will also use stem bark, and some would just use resin. Some would use resinous wood. So there are so many terms referring to the same thing, so which should have been standardized to have a better and effective research publication. And there are also other issues like unresourced species names. Sometimes the same name refers to the different name refers to the same species. So the recommendation is that to always go back to the authoritative websites and also the horticulturists to actually authorize the species that we are using. And following that, we recommend some sort of standard reporting or preferred reporting guideline, or at least share some minimum information in each publication. So that this would actually benefit the Agarwood research vicinity. For example, some of us may be very familiar with PRISMA, which is the preferred reporting item for systematic reviews and meta-analysis. So when everyone follows the same guideline, so that would help all the researchers provide the minimum information so that the research can be conducted in a systematic manner. So now moving forward, what is the future for Agarwood research as well as natural product research? So as you can see from this slide now, a continuum of ethnopharmacology to drug discovery. As mentioned earlier, Agarwood has been used in many types of different traditional medicine, from China to India, and of course as well in our Malay culture. So ethnopharmacology simply defined as pharmaceuticals that are actually originated from the traditional medicine. So in this paper here, written by the expert in the area, Prof. Michael Hendrick is one of the experts in this area, ethnopharmacology. So they are looking into this neuroscience, nutritional neuroscience, neuropharmacology, and food science. So when people in areas similar to me, the natural products, the traditional medicine, so in the future, we are looking into the integration of this ethnopharmacology to drug discovery in these three main areas. So this actually warrants further systematic and scientific approach to look into these different areas with, of course, the more up-to-date methods and approaches. And the second way forward is a term called Eco-Pharmacognosy Approach. So we may not be very familiar with this approach, but we may be familiar with the term food security, which is very important now. But there is also actually another term very similar to this, which is medicine security. And when we talk about medicine, which actually originated from the natural products or resources or plants, which majority of them are either extracted from the plants or at least being inspired from the plants. So we are talking about sustainable medicine. So that is why it's very important to have the to save the traditional educational knowledge, the TEK that we got from the forest, from the people dwelling in the forest, the pribumi or the orang asli in the country. So according to this paper written by Prof. Cordell, also an expert in this area, akin to this food security concept, the eco-pharmacognosy approach sets to achieve medicine security. So for Agarwood, because it is one of the natural product resources, it shall benefit from this approach as well. Okay, the last part of this way forward is actually coming back to roots. So this is the missing link in the slide that I've shown before. As I've shared before, we started with zero waste, waste to wealth, a bit of SDGs, a bit of circular economy, but there was no Maqasid-based approach back then. But Alhamdulillah, with our university advocating Maqasid-based, Maqasid-ShareAlike-based approaches, we are also coming back to roots, I would say, coming back to roots and moving forward. So we started to deliberately look into this, and Alhamdulillah, in late 2021, when we tried to explore this concept, we had the opportunity as well to share this with colleagues from Brunei Darussalam, from the University of Islam Sultan Sharif Ali in Brunei Darussalam in this conference. So we tried to map back what we have done thus far with Maqasid Sharia, as well as Maqasid Al-Quran. So for us, at least for me, I think that is what we should have been doing right well before, but it was not there. But Alhamdulillah, we're given the opportunity to do it now. Because why? Because we are Muslim scientists, so we have to be guided in the correct way. And the second why is because we are from the IUM, so we must do this. If not, who else would be doing this? So it's very late when we come back to Maqasid-based, but Alhamdulillah, we are doing it now with the help from our colleagues from various kuliah, where we break the silo again as advocated by the university. We gain something good, Alhamdulillah. So with that, perhaps the last part, which is not in the slide, is moving forward in mapping our research to 1010 My SCIE, which is very important for all researchers in the country now. So finally, I would like to thank all the team members, the current one, as you can see here, as well as the past members and everyone who has been in the journey. It's a very wonderful journey actually for the 10 years, and we hope that we can work together for many years to come. And finally, I would just like to share pantun, which to show you that actually Gaharu has been there in our Malay culture for many centuries, and it's just our responsibility and amanah to uphold it with our culture and use it as the best as we can as a khalifah. So allow me to read this pantun. Sungguh harum bau Gaharu tidaklah sama dengan cendana. Sungguh banyak orang yang baru, tidaklah sama orang yang lama. Naik perahu belayar ke seberang, menuju belukar menebang kayu. Bukan tak tahu zaman sekarang, Gaharu dibakar, kemenyan berbau. Cari galah kayu di hutan, singgah menetap cendana Gaharu. Minta doa mudah-mudahan, boleh rezeki datang selalu. Terima kasih and thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Prof, for your amazing sharing and presentation. I believe I should reply back the pantun from you. Okay. But I'm so sorry if there is Bahasa Melayu teachers out there. If the rhyme is sound weird. Okay, so pokok gaharu, pokok cendana, sedap dibuat minyak wangi. Wahai Prof Yumi yang bijaksana, insya-Allah kita akan jemput lagi.
InsyaAllah, terima kasih.
Thank you so much, Prof. It is indeed very interesting and amazing sharing, because normally we know Gaharu only synonym with perfume. Okay, I think minyak atar also come from Gaharu. Is that correct, Doctor?
One of the ingredients could come from Gaharu.
Oh, okay. So from this sharing, I can conclude that many bioprospecting in Gaharu. Okay. So thank you so much. It is definitely new knowledge for me, and I believe for some people out there who are not in this field. Thank you so much, Prof.
Thank you.
Okay, ladies and gentlemen, coming up next is our second panelist for today, Associate Professor Dr. Muhammad Ibrahim. Assalamualaikum, Doctor. Can you hear me? Okay, ladies and gentlemen, let me read out the biodata of our second panelist. Associate Professor Dr. Muhammad Ibrahim received his Bachelor of Technology, Specialist in Food from University Science Malaysia in 2000, and then he continued his master degree in food processing and preservation from the same university in year 2004. In year 2010, he obtained his PhD in nutritional biochemistry from University Putra Malaysia. Associate Prof. Dr. Muhammad Ibrahim areas of specialization are food sciences and nutrition, nutritional biochemistry, food sciences, food processing and product development, and sensory evaluation. He is currently the Deputy Dean of Postgraduate Responsible Research and Innovation at Kuliah of Allied Health Sciences. Associate Professor is active in community engagement and knowledge transfer project. Among his research that involve with 6 small and medium enterprise is Kelulut propolis extraction made easy training of Trigona propolis extraction using easy and economical method for local apiculture, small and medium enterprises, or known as SMEs. Okay, now ladies and gentlemen, presenting to you our Associate Professor Dr. Muhammad Ibrahim to present his research project entitled Simple Method of Propolis Kelulut Extraction. Please welcome, Doctor.
Okay, thank you very much, our chairperson for today. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala ashrafil anbiya'i wal mursalin. Sayyidina Muhammadin wa ala alihi wa sahbihi ajma'in. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Wasaddid lisani wahdi qalbi bihaqi Muhammadin sallallahu alaihi wasallam. My dear respected program committee of the programs and all the brothers and sisters, today I would just want to share a simple thought on how we gonna to benefit the propolis kelulut, and then normally this particular propolis has been a byproducts of the kelulut honey, and then it is not being fully utilized. My slides today will be in Malay so that it will benefit our SMEs in Malaysia, and also all our brothers and sisters in Malaysia as well that are going to benefit from this particular thought, inshallah. So what I'm going to share with you today is about the compositions of the propolis kelulut, and then some of the health benefits of propolis kelulut, and what we have done related to the research related to the propolis kelulut in IUM Kuantan, and also I would like to share with everybody how we can prepare the propolis kelulut to becoming a good samples, and then we're going to further extract this particular propolis kelulut to make use of this particular extract in our daily use. And at the end, what is the best method to make sure that this particular extract can be sustained longer, and then it's going to be quality, to ensure the quality of the extract are being preserved accordingly. Okay, as far as we all know, there are lots of study and that has been conducted related to the propolis kelulut. What I'm showing now is the chemical compositions of the propolis kelulut. It contains carbohydrate, it contains amino acids, vitamins, phenolic acids, flavonoids, and others antioxidant compound that has been studied on. So if you look at the chemical compositions of the propolis kelulut, it varies because of the geographical factors, and then the excess of the source of plants that has many what we call it as availability surrounding the hive of the propolis hive, beehive, and also it related also with the seasons, and then many of this particular active compound has been shown to have complex mechanism with the propolis, how this particular propolis act as antivirus, antibacterial, anti-mold, and also providing some antioxidant effects in the scientific studies. So this is the examples of the study that's been conducted on the physicochemical composition of methanolic extract propolis of the stingless bee Kelulut. Kelulut is stingless bee in terms of moisture content, ash content, and if you look also, it contains of the high fibers and also the carbohydrates. Other than that, also, for example, it contains the flavonoids, terpenoids, saponins, all these particular antioxidant polyphenols, and then not to forget, it is also contained the essential oil as well. So there are a lot of studies that has been conducted with related to the health benefits of propolis kelulut, including anti-parasite, anti-ulcer, and then also the product from the Colgate as well. They use this is the propolis, not from the propolis kelulut, to be used as an antibacterial for the mouth gargle, toothpaste. Other than that, in the gynecology aspects, for example, being the propolis kelulut being used as a anti-mole, as a biofilm, dermatology, for example, related to the diabetic foot ulcers, oncology field, breast anti-cancer, breast anti-cancer activities, and so on. So if you look at the overall of the health-related propolis kelulut, previously it has been used traditionally to have their effect on the sore throat, and then to relieve the symptoms of flu symptoms, and as well as in the any related oxidative diseases, oxidative stress related diseases as well. And then also in the ulcers treatment, and also in wound healing and process. So there are lots of what we call it as a product hiliran for the propolis kelulut, byproducts of propolis kelulut that has been produced in pharmaceutical industry, for example, lotions, and then topical creams, in terms of gargle for the mouth, and then also it has been used also as a topical in the ubat, kita kata apa, ubat penyembuhan luka. And then also in terms of in the food products development also, this particular propolis kelulut has been used as a preservative in the food product to prolong the shelf life of the products, and then being used in the candies, and then in the production of chocolate bar, for example. Other than that, in the cosmetic product, for example, anti-acne creams, skin lotion, shampoo, and that has been used as well on the propolis kelulut. So not to forget, we in IIUM Kuantan has embarked in this particular research for many years as well, but not as long as Prof Yumi as 10 years of the study in Agarwood, but not to forget also we are already studied this around 4 to 5 years. This is four studies that has been conducted recently to make sure that the product that has been produced by our lab are sufficient to address the community and also to address a certain point of to help the SME and to come up with a good product. So the first study that has been conducted is antioxidant activity of aqueous extracts of the Trigona species Propolis. And then this particular study, we are using the computer software to optimize the antioxidant produced from this particular extract, what we call it as a response surface methodology. And then the second one, we try our best to optimize the phenolic compound in the aqueous extract and antioxidant activity of Trigona honey and also Propolis honey. Trigona Propolis, similar, the combinations. To see how this particular combination, the honey, Kelulut honey, and as well as the Propolis Kelulut, have the impact on producing the combinations of propolis mixture with the Trigona honey and the mixture that high in polyphenolic compound and antioxidant activity, also using the response surface methodology. And then the third study that's been conducted here also to optimize the extraction temperature, time on phenolic compound and antioxidant activity of the Malaysian Propolis Trigona species aqueous extract using surface response surface methodology as well. So this in the third study, we are trying to optimize the temperature, the time that needed to have the highest antioxidant activity of the extract. And at the lastly, this particular study embarked into the to see the efficacy study on the wound healing, using the combinations of Trigona honey as well as Trigona honey propolis, Trigona propolis, and its combinations as well. So this is our research team.
We do hope that this particular research can be further studied among our human population, inshallah. Now we are trying on the animal first, and then inshallah, if the result is positive, we will further study in the human clinical trial, inshallah.
So hopefully our brothers and sisters enjoy the video, just to show everybody about the effectiveness of our study on the wound healing process that has been used propolis extract as a medium to further increase the wound healing process. So there are one chat there that how to get the propolis extract. Actually, we are in the process of asking the company to the SME to pack for us. So in the near future, inshallah, I think the product will be available as well in the market, inshallah. So for those who are interested to test that particular propolis extract, can have a contact with me lah. Inshallah, I will provide to you in a very cheap price lah. So I just want to share here about the study that has been conducted with related this particular extraction process of the propolis on the effects of temperature and the incubation time to have the highest antioxidant propolis antioxidant extracts. So we start our extraction condition with the temperature ranging between 30 to 60 degrees Celsius, and then the time of extraction range between 24 hours to 72 hours. So this is our key findings, and the optimum extraction condition to produce the highest antioxidant yield is the optimum extraction temperature at 43.75 degrees Celsius, and optimal extraction time is 52.85 hours. It is around 2 days of extractions. And then when we look at the experimental values are very close to the predicted values that being produced by the response surface methodology. So this is our findings, whereby we can see here the response surface plot of the temperature of extractions, temperature and extraction time, the effects on the total phenolics, the total phenolic compound, on the total flavonoid compound, and also on the antioxidant activity. We conduct the DPPH testings, ABTS testing, and also the FRAP testings of the trigonal propolis samples. So this is our findings as we come to the conclusion that the combinations of time and temperature will produce the a good total phenolic compound, total flavonoid compound, and a few antioxidant assay that has been conducted to show that the time and the effects of time and also temperature on these particular antioxidants. So this particular study that has been published in Malaysian Journal of Analytical Sciences. And then the second study that has been completed as well is the effects of the combinations of honey and Kelulut propolis on the antioxidants. So this is our testing combination amounts. Honey range between 15 to 16.5 gram, propolis range between 13.5 to 15.5 gram. Because in this particular trial, we try to optimize the antioxidant that we have from the Kelulut honey as well as the propolis honey to becoming and what we call it as a mixture that can be used later on in the food preparation, in the pharmaceutical area, in the cosmetic area. So this is the key findings. Our key finding is the optimum combination to produce the highest antioxidant yield is optimum amount of honey around 50.43%, and optimum amount propolis should be used at the almost 50%. So this is the effects of the combinations of honey and also propolis, kelulut on the antioxidant parameters. So as we can see here, the combination of honey and propolis around 50% each of contribute to have the antioxidant total phenolic contents of 152 milligram galic acid equivalent in a 100 gram of the combinations. And also it poses a good antioxidant activity as compared to others, propolis and also honey. So we are already published this particular findings also in international food research journals. So the last one is we're going to look at the effects of the propolis kelulut topical on the process of wound healing in guinea pigs. So if as we can see here, we have 1, 2, 3, 4, 5. Five groups of guinea pig that has been applied with the positive control group, whereby in this particular group we are using a common topical cream for wound healing. And then in experimental we use topical propolis. In group 4 we use oral garbage propolis, and then the group 5 we use the topical and oral propolis combination. And then we have a negative control group whereby the wound being washed out with a normal saline. So as we can see here, regardless of what treatment that we use, whether topical propolis, oral garbage propolis, or topical oral propolis, it's shown a comparable or better than the from the positive control, which is we use as a common topical cream for the wound healing available in the market. So now we are at our main focus now on the simple method how we can prepare the propolis kelulut. We can do it in our home actually. If you have the hive of kelulut in your house, and then you are using the honey for the consumption, not to forget that the propolis also, don't throw away the propolis. We can make use of that particular propolis. So in this particular simple method to prepare a propolis kelulut, we can take the propolis, raw propolis, and then we can do the process of the extractions, and then in this particular extraction process, we're going to remove all the dirt, all the dirties on that particular propolis. And then also propolis, we can get the concentrated and pure propolis from this particular process. So what we have to have is the blenders, mechanical blenders, and then if you don't have, and then we have to have a balance as well, balance. And then if you have to prepare also the vortex, and then a convection oven. If you don't have the centrifuge machine also, never mind, we can use also the gravimetric filtrations. Don't worry, we can always use what we have in our house. And then for the betterment of the extraction, we should have also rotary evaporators, and then others equipments, beaker, parafilm. So this is our way of doing of the preparations of propolis kelulut sample. So first we have to clean the propolis from the all the dirties that are if we get this particular propolis from someone else, and they might not store the propolis carefully, so that there are lots of dirt on the propolis. We have to wash it out, and then we have to cut all this particular propolis into the small size, and then we have to crush the propolis using the crusher, and then we can blend this particular propolis using the mechanical blender, and then it should be coming in a form of powder, and then weigh this particular propolis powder, and then insert the propolis into the beaker, and then we can mix this particular propolis powder with water, and then make it the ratio of this particular propolis powder to the water 1:10, and then we have to wrap that particular beaker with aluminium foil, and then close that particular beaker with a parafilm. So, and then we can put this particular mix into the oven at the temperature of 49 degrees Celsius for 24 hours or more than that. So after that, we take out the samples and leave it cool, and then we can transfer the propolis mixture into the stirrer. So we have to centrifuge this particular mixture for about 5 minutes if we have the centrifuge at our settings. If not, we can filter it, but it will take a longer time, and then we can transfer that particular extract to the another beakers, and then we should discard all the wax and the waste, and then we have to repeat this particular process again and again to have a lot of extract. And then this particular extraction, pure extraction propolis should be concentrated. Concentrated using the rotary evaporator at this particular settings. So this is the pictures showing how we are using all the materials that we have to produce the kelulut extract. This is the picture of raw propolis extract. We have to chop it down and then crush it into a small pieces and put into the blender and make it into the powder forms. And then we have to measure and then put the ratio of powders and the waters, and then put it in the ovens for quite some times, and then we have the crude fiber crude propolis kelulut propolis extracts. And then also you can have a very quick method of preservations. You want to keep your extract freshly and then not degrade and whatnot, and then you have to make sure that after you collect all the extract from all those process, this particular propolis should be covered from the exposure, from the light, heat. If not, it will degrade. So it is suggested to be stored to store it at minus 20 degrees Celsius until you want to further use this particular crude extracts. And then after you after the what we call it, concentrated process, after the evaporation process, you can store it at 4 degrees Celsius until we want to convert it to further process into the products, final product. And then this is my books that has been published with the library, the library on the Propolis extraction module that are available in our library. And for those who are interested with this particular project, I'm going to conduct the this particular projects or this particular project on the Propolis extraction module, inshallah after Hari Raya. After Hari Raya, we're going to have the course with the SME that has been decided in the KTP grant, and also I would like to invite others as well who that are interested to join our module Propolis extraction module, inshallah. And then with that, I think that's it from me. I pass back to moderator to continue the session. Thank you very much.
Okay, thank you so much, Prof. It is indeed a very informative sharing. Normally, I can say that we usually use about the benefit of honey, tualang honey, manuka honey, kelulut honey. But from your presentation, we can see how beneficial the propolis is. And I want to relate something that in Al-Quran, there is also one surah named An-Nahl. That's mean the bee. So we can see how beneficial the bee is. So from honey and the propolis that come from bee really give many benefits to humans. Okay, so thank you so much, Prof. Okay, now, ladies and gentlemen, we have come to the long-awaited session. It is Q&A session. So you may write your question in the chat box, and I will pass the question to the respective panelists. Okay, I see there is a few questions. Okay, so we start. Okay, so there is one question from Brother Muhammad Rizwan. Where does the best Agarwood come from? Okay, over to you, Prof. Yumi.
Okay, thank you. Thank you, Brother Rizwan, for the question. So the answer would be depending on who is asking. Why I'm saying that? Because some of the people from the Arab countries, they really like the Agarwood from here, from our country, from our nation. So they would say Aquarium Malacensis would be the best source. But for people in Japan, for example, the best agarwood would come from Vietnam. So they are from different species. They have different aroma. So that's perhaps the short answer for that.
Thank you, Prof. Okay, next. Assalamualaikum. Mana nak dapat propolis kelulut ni? Boleh order ke? Okay, that's from Sister Zuraini. Okay, Dr. Muhammad.
Okay, currently we are not producing in mass in mass production. I would say we are in a process of I'm not sure whether we are going to patent it, but we are still in the discussion with the innovation and commercialization unit what to do with that particular products lah, because we are already optimize it, and then we're gonna do I'm now planning to further study on the clinical trial, and then we are already approaching the orthopedics and also the internal medicine department in our Susmet hospital. But for those who want to have a try, we do provide. They can contact me lah. We do provide that extraction.
Okay, thank you so much, Doctor. Okay, so next, what are the other uses of agarwood beside perfume? Cosmetic products? Okay, Prof.
Okay, yes, as we know, perfume is the main products, the fragrance industry, but it's also being used as ingredients in cosmetic products, and less popular, but there is in food. So we're talking about agarwood here, we are actually not just referring to the aromatic resin, we're also talking about the different raw materials. The leaf is being used as tea. It's also being included in biscuits, in noodles, dried noodles, mee. And apart from that, agarwood as a whole has also been into the sector of agro-ecotourism. So if you're aware, in Gopeng, there is one Gaharu Valley called Hoga Gaharu Valley in Gopeng, where you can go there and have, it's like a resort where there are a lot of Gaharu trees. You can go jogging there, the breakfast there. So that's another sector. And other less common products, perhaps not in our Malay culture, but it is very much in Chinese culture, Japanese culture. They use this agarwood trunk to do sculpture, carvings or sculpture. So it's very expensive in their culture. So those are some of the things. And perhaps the one that we are sharing today, the health beneficial effects, is the most challenging one, because you've seen it to be used in many different traditional medicines, but unfortunately there is none really one modern medicine based on agarwood. So that's what we are doing as researchers now.
Okay, thank you so much, Prof. Okay, the next question from Madam Hashimah. Dr. Muhammad, the color of Kelulut varies in the market, and I understand it depends on what the bee consume. Does the color, sorry, I don't see. Does the color affect the effectiveness of the wood healing?
So in our study, we don't go for the specific apa ni kita panggil apa? Species of the Kelulut. For example, we have Lavisat, we have Itama, we have a lot of different species. In Malaysia, we have 38 to 40 species. So these different species produce different color of honey. For example, in this particular case, and if now I'm talking about the propolis, even the question on the honey itself, different species produce different color of honey. So what we are doing here is to standardize this particular propolis honey or the propolis honey, brother. Kelulut honey or propolis kelulut to have a certain kita kata apa bahasa mudah dia we use a reference materials to standardize the bioactive compound that inside of our extract or in our honey itself, and then we are now collaborating with USM to come up with one standard that can be used in the clinical trial. The problem with the honey and its propolis ini, we cannot guarantee the stability of the compound inside the propolis extract or the honey itself, because it is high in moisture content. So this is very challenging lah, challenging in the standardization process of bioactive compound inside a product. That's why when we convert the honey itself, or we convert the any extract, the very challenge is to maintain the quality of bioactive compound, and then make sure that bioactive compound act as we study in the fundamental study. This is very challenging. But if you want to have a different color of Kelulut honey, you can go to Kelantan. There are one what we call it as it is one resort that has been dedicated to have something like eco resort eco resort just beside the University of Science Malaysia Kubang Kerian. So you can visit there. There are lots of different color of honey, Kelulut honey. They use different types of fruit trees, and then this particular fruit trees giving a significant colors to the honey that being produced. For example, over there they sell about 500 milligram, half a kilo of Kelulut honey at the price of 10,000. Because this particular Kelulut honey being produced by the this particular honey consume mangosteen extracts, and then the colors is slightly red, in reddish color, and the taste is very nice. So if you have a time, you can go there lah. So this is the panjang pula lah.
Okay.
I think, Sister Moderator, what Dr. Muhammad was saying just now is generally true for all natural products. You want to make sure the bioactivities are there when you develop products for the consumers. Yeah.
Okay. So next question. How many species of agarwood and most expensive agarwood from which country and which species? Okay, Prof.
Okay, Madam Farika, this is actually a similar answer to the previous question. But in general, our Aquilara malacensis from Malaysia is so-called the best species, producing the most expensive agarwood. And as I said just now, from the Oriental nations, they perhaps prefer the agarwood from Vietnam and Cambodia.
Okay, thank you so much. The next question. Okay, from Sister Haslina. Is Kelulut bee harmful? Is Kelulut bee harmful?
You can come to IUM Kuantan. We have here in what we call it Taman Insan Sejahtera at IUM. On the top of the hill, we have around 50 hives there. You can visit us, and then you can. In fact, in Gombak campus we have as well. The group from the Gombak campus as well has working on this particular Kelulut. But if you are interested, come to Kuantan, and then Kelulut is not harmful lah. That's why the name is stingless bee. The bee without sting. But even having said that, also there are a few species that are very aggressive, and they will produce a special special compound that when they even though stingless, but they can bite as well. So it's going to introduce some minor inflammations lah if you have that. But basically it's not harmful lah. Very easy to manage, and then very friendly.
Okay, thank you, Doctor. Maybe one day we should organize one program. We go there, visit the bee. Okay, Prof Yumi, maybe we can see the biodiversity.
Interesting.
Yes. Yes, interesting. Okay. Okay, so next question from Brother Muhammad Rizwan. How long does agarwood takes to mature?
Okay, in a plantation, normally 5 to 7 years of life cycle. In 2 to 3 years, they will start injecting, and then they will allow the plant to grow and produce the agarwood before being harvested. But in the nature, in the forest, generally the longer it's there, the higher the quality of the resin. It goes back to the previous question actually. The more expensive agarwood is actually from the nature. The perception is like that. But of course, we cannot go and harvest all the trees in the forest. So that's why we do it in the plantation. So it's 5 to 7 years.
Oh, okay. So I want to add one question related to this. So, do we need like license for to get the agarwood from the forest? Like, do we
Yes,
need special
indeed.
license? Yes.
Yes, there's a special license because as you know, the products from the forest, only the pribumi, the orang asli, has the rights to go and use it for their daily living. But unfortunately, that is where the biopiracy comes in. So I introduced to you the bioprospecting. I did not really mention about biopiracy, but that is opposite of bioprospecting. So what actually happens in agarwood industry is actually people, not the pribumi, they so some sort like make use of our pribumi. So they what is it called? Poaching, illegal harvesting. So they take out the Gaharu tree from the forest and they smuggle it. And in our country, the real stories, usually it goes through our neighboring countries for smuggling. So that's a scenario that's still happening now, although we are talking about the so-called bioprospecting.
Okay. Thank you so much, Prof. Okay, the next question. Can propolis cure COVID? Okay, doctor, this is our issue. issue
I
nowadays.
do not know. I do not know. I do not know. The answer is I do not know because this one I can pass to my colleague here lah, biomedical or Prof Yumi. In fact, can do the research because infectious disease is not my field. And then so far I think that there are some orang kata apa ni studies using the not the propolis as well, the honey itself on the effectiveness in helping the COVID people to recover back lah. There are I saw lah a few studies. Tak ada masalah. Tapi for me I do not know. Because this is out of my specialization lah.
Okay. Okay. Yes.
New
Normally.
research.
Yes. Normally.
New research.
Yes. Normally nowadays we get message like this in WhatsApp. Eat honey, madu. So maybe there is one specific research, okay, regarding this matter.
But it may help from the therapeutic diet perspective, because I'm from the dietetic department. From the diet perspective, if you consume kita kata apa ni significant amounts of bioactive compound that will facilitate your immune system, for sure. Not only propolis, not only honey, but a correct way of eating punya practices lah.
Okay. Thank you so much. Thank you so much, Dr. Muhammad. Okay, next question. What are the halal products based on agarwood available in the market now? Okay, Prof. Yumi.
Okay, thank you for the question, Sister Aslina. Available halal products on agarwood, there are a lot of cosmetic products out there. Some that I mentioned, the food products, are all halal, inshallah. I mean, please check. That's our job as consumers. Check the halal status of the products.
Okay, thank you so much. Prof Yumi, I want to ask one thing, like related to the product. Is it do you know Kemenyan? Do they burn a gaharu in Kemenyan? because I suddenly
like the pantun that we read just now, ada kemenyan, ada gaharu kan? So
Yes.
that actually relates well to our culture. We normally we are very familiar with the bau of kemenyan,
Yes.
right? In many
Yeah.
of our rituals, religious as well, the cultural rituals. But we don't normally burn gaharu. Oud is something more related to the Arabic tradition. So
Mm.
sometimes actually we do put gaharu in a small amount,
Oh,
but
okay.
the smell is actually a bit different if you really know how to differentiate them.
Okay.
But in Malay culture, I would say we do not really burn oud. But in
Oh,
Arab culture, yes, that's their daily routine.
okay. Thank you so much, Prof. There is one question from Sister Haslina. Apakah spesies-spesies bunga yang kelulut suka hinggap? Okay, Dr. Muhammad.
Generally semua bunga sebenarnya, sebab apa ni, kelulut ni being used as a pollinating agent as well. Pollinating agent, agent pendebungaan, agent pendebungaan, pollinating agent. Tu Siti Azizah, Sister Siti Azizah kata min house camp Kubang Kerian. That's the place that I mentioned earlier, min house camp Kubang Kerian. You can go there, very nice place. So spesies bunga ni normally kebanyakan many of the kelulut farmers dia akan tanam bunga air mata pengantin lah. Bunga air mata pengantin. I do not know why that particular bunga being named as air mata pengantin. I'm not sure whether the tears is a sad tears or enjoyable tears during the wedding. I do not know, but this particular flowers can easily grown lah, easily grown. It is a sort of kita kata bunga air mata pengantin. And then for your information as well, I'm been coaching the Persatuan Kelulut Pahang more than 10 years, I think. Persatuan Kelulut Pahang more than 10 years. And then in fact in the Bukit Goh we have one Kelulut farm. One Kelulut farm being sponsored by Yayasan Hasanah. Being sponsored by Yayasan Hasanah. We name it as a Kampung Kelulut. I think that over there we already have around 400 hive there, and then I've been coaching them to come out with many products for the into the market as well mana maju ke. But for the propolis extract, we are still in the process of establishing the correct method, and then how to store it properly, and then how to make sure that the quality being kept at maximum quality lah. So species bunga actually, any species bunga actually, but this particular kelulut are really enjoying when they are surrounded by this particular air mata bunga pengantin. Ini bunga air mata pengantin. Easy to be grown lah.
Okay. Thank you so much. Okay, thank you so much, Doctor. So, Doctor, maybe different kind of flower give different kind of taste in the honey, right, Doctor? Because I have tried madu kelulut once. It's like sour. It's not like sweet. So, is it the correct taste?
Because if you look at the pH of this particular madu kelulut, is slightly acidic as compared to others. Slightly acidic as compared to others honey as well. So the preservation technique is very important. When we harvest the honey from their hive, so that it has to undergo a proper preservation techniques to make sure that we stop the degradations of certain compound inside the honey, to make sure that they are not producing too acidic compound inside the honey lah. So yang ini kena lecture lain lah.
Okay. Okay, next question. Okay, from Sister Farika. Is Agarwood oil safe to use directly on the skin? Okay, Prof. Yumi.
Okay, as of any other natural products, when we said when we talk about safety, there's always a level of safety. So what I would recommend is, if we were to use agarwood oil, or we call the essential of any plants, please go for a trusted brand. I would not say any brands, it's not a promotional session, but what is it? Go for a trusted brand which have tested the level of toxicity, the level of safety, which also will recommend how much to use, how to use it, how many times, so that everyone is safe.
Okay. Okay, thank you so much. So next question. Oh, from Sister Farika also. So how much cost involved to set up Kelulut bee farm? Is it affordable? Okay, Dr. Muhammad.
Kalau farm itu we have tried it at at a cost of dekat Bukit Queen itu we install around 50 hive that are stable hive, what we call it, stable hive. 50, it is cost around 100,000, including all the fences, all everything, because you cannot get the Kelulut bee hive, a stable hive, at the very cheap one as compared to the previous years lah. Previous years I bought from our indigenous people, maknanya orang asli kita, at 30 or 50 ringgit per hive previously. I go there, I want to teach them how to develop a stable Kelulut farm, but they sell all to me. They sell all. They say to me, you take it and you do it yourself. We sell it at a very cheap price, so I take all. So I think you can do it at your backyard for a start. And make sure that you train yourself first, and before you go to the orang kata apa ni, a farm punya setup lah. A farm punya setup. You are the feeling good kind of kita panggil feeling good kind of attraction dengan Kelulut ini. So if you can maintain a few months at your backyards, and then you can affordable. If Sister Farika really want to embark in this particular Kelulut punya business, kita ada model, in fact. Kita ada model, kita dah propose dah sebenarnya yang this particular model to the Ministry of MAFI, is it? Sekarang ni MAFI, Food Agriculture and Industry. In fact, saya dah propose dah last 2 years, saya rasa, tapi tak tahu dah proposal jadi apa.
Okay, thank you, Doctor. So, Doctor, the hive for the Kelulut bee is like we got from tree block, right? Is that correct? The hive for the Kelulut bee is from tree, right? Like.
Yes. Previously they cut the tree. Now we are having a different method to make sure that this particular honey can be breed in a farm. Different person use different method.
Okay.
What we call it as to have a new colony of the bee lah. It's not necessary you cut all the log in the forest. You cut the tree to take the log from the tree. We don't do that anymore.
Okay.
There are lots of technology being developed by Mardi, recently being developed by Sirim, in fact, working on with Mardi as well.
Okay. Thank you so much, Doctor. Thank you so much. The next question from Brother Mazaimi. Kenapa rasa madu kelulut terlalu berbeza dengan madu lebah sedangkan ianya datang dari bunga yang sama?
Okay. Sebab dia diproses oleh orang yang berbeza. Different bee produce different enzyme.
Okay.
Different bee produce from different enzyme. That's why in the process in a kelulut in a kelulut punya stomach itu dia slightly acidic as compared to other bee. Slightly acidic. So it produce more acidic compound lah.
Okay.
It's not just based on the what type of flower that they use to take the nectars, to take the honey and whatnot, but it is because of the processing in their stomach as well.
Okay. Okay, thank you so much, Doctor. Next question is from Brother Muhammad Aslam. May I know what is the purpose of addressing Makkosi and Halalan Tayban elements in researching Agarwood products? Why it is necessary? Thank you. Okay.
Thank you, brother. It's a big question for us because, as I said just now, we are just embarking, coming back to the roots. So actually the answer is going back to one, when we do Maqasid-based bioprospecting, inherently, inshallah, we'll come out at the end product is the Halal and Tayyiban products that we would want. Some of the Halal and Tayyiban elements we are looking into as Halal built-in approach, Halal science. So whenever we do things, we go back to the guidance from the Quran and Sunnah. So inshallah, we'll be guided as Muslim scientists. So that's the main thing. In fact, in Kelulut research as well, I think all of those are bioprospecting, Maqasid-based. It's because we are Muslims, we need to be guided. And what is the best guidance that we have is Quran and Sunnah. And how to make sure we are in that line is to go by and follow this Maqasid Sharia based. Thank you.
Okay, thank you so much, Prof. Okay, so what I can conclude that related to what Dr. Muhammad said, like nowadays there is no cutting tree. I think that is the additional element of Maqasid Sharia, right? So
yes,
everything in our life is crucial to have Maqasid Sharia elements. Okay, right, Prof?
Correct, yes.
Okay. Okay, so let me see if you have another questions. Okay, so here's one. What are the other uses of Agarwood besides perfume and cosmetic product? I think we already answered this one.
Yeah, we did. Yeah, that one already done, right?
Already done. So I think that's all for our...
I think Sister Zara, perhaps there's one question on the quality of Agarwood. I thought I saw it somewhere just now.
Okay, let me check first.
How we check the quality of Agarwood? Yes, from Brother Rizwan.
Yes, the quality of
scientifically, yes, we do check quality of Agarwood. It relates to the grading as well. The higher the grade, the more expensive it is. So in the lab, there are two things that we normally check for. Number one is the oil. Number two is the resin itself. So the one that we use for the fumes, the oil. So in the lab, we use an equipment called GCMS, gas chromatography mass spectrometry. And in fact, at INHART lab, we do provide these services. In Kuliah Engineering lab also, they do have this service. So we do have actually contract research, not contract research, contract service coming from several industries that asked us, requested us to check. On the resin also, we do have scientific methods to check this quality. However, I must say this, in the Gaharu industry, especially in our country, the main players are all the SMEs or the farmers. So they do not really have the big amount of money to go for these scientific methods. And another factor is that the buyers, especially from the Arabic countries, they are still using the human way of checking. If they use their nose, menghidu. So they are trained to to smell which one is of a quality, thus a higher grade, and vice versa. So that's what's happening in the industry. Not many are using the analytical equipments, and of course, if you know that there are also in the market the eNose. Okay, actually, some of our researchers also developed that, the eNose, to check the quality and the grading. Unfortunately, it is not also being used widely in the industry.
Okay.
Yeah, thank you.
Thank you so much, Prof. Yumi. I think that's the last question.
Yeah.
Yes, I think that the last question for today. So, okay. So thank you to all participants for your question and support. Okay, so we already shared with you in the chat box both email address for both speakers. So if you have anything to ask them or you are interested to collaborate with both our speakers today, so you are feel free to email them. Okay, well, ladies and gentlemen, that's wrap up. Okay, well, that's wrap up our session for today. On behalf of Dar Al Hikmah Library IIUM, we would like to thank you all for making time in your busy schedule to join us for this session. Okay, before we end, do not forget to subscribe to our channel and click the notification bell so you will not miss out any interesting event or content uploaded here. Once again, we would like to express our heartfelt gratitude to the respected panelists and all participants. Okay, thank you so much, Prof. I think we get a lot of knowledge today regarding honey and Gaharu. Okay, then we would also like to apologize for any shortcoming and inconveniences. Hope to meet you again in the next session and wish you a pleasant day and stay safe wherever you are. Let's end our session with recitation of Tasbih Kafarah and Suratul As. With that, wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. And thank you.
Waalaikumsalam.